So what I want to show today and to talk about is the interaction of two of the most fundamental parts of gene regulation, or how the cell controls which genes are expressed and then actually contribute to cell function. And you can see this in the video here already. So you have on the left-hand side a gene regulatory control element of the genome, or an enhancer, or in this case, a super enhancer. And on the right-hand side, you have a gene that is interacting with the super enhancer that has a cluster associated with it. So we're going to really arrive at the understanding of all the components that are inside of this video, but that comes, of course, later in the talk. To give you a little bit of an orientation, and also to place this in the theme of condensation and phase separation, I would refer to sort of this very simplified, comprehensive summary of some of the things that we learned about how you control and carry out transcription in different spatial compartments that are established by condensation. So what I mean by this is that if you look at the control of transcription, you have elements such as enhancers here that control transcription by interacting with promoter regions in the genome. And here you have transcription factors that bind transcription machinery, and importantly, RNA polymerase II that needs to go onto the gene, or actually the promoter region initially, and then goes into elongation. And this whole aspect of controlling what is transcribed and what isn't takes place, or at least that's the emerging theory now, takes place in so-called promoter condensates. These contain high levels of recruited RNA polymerase II. This has a specific phosphorylation mark, serine 5 phosphorylation of the C-terminal domain. And then here it's decided what is being transcribed. Once the Pol2 transitions into actual transcript elongation, so here a production of RNA, this takes place in a different condensate that is spatially distinct. So this is where really the RNA polymerase is producing the RNA transcript. And you can also find an additional, so it doesn't lose this phosphorylation mark, but it additionally gets a serine 2 phosphorylation in the C-terminal domain. Now, for completeness sake, I should say that actually only about 10% of the Pol2 maximally goes over here, and about 90% is released and retains this phosphorylation mark and becomes part of this condensate material on the left-hand side here. So that's, in a sense, the general picture that is emerging, how you can spatially compartmentalize the control and then the actual process of transcription. And this becomes extremely prominent if you go into embryonic or pluripotent cells. So our favorite setting to do this is the zebrafish embryo. You can see it sketched on the left-hand side here in the pluripotent stage of development, the sphere stage. It's transparent, it's an embryo, so it's probably quite natural and healthy. Hasn't been in cell culture for years. And if you look inside here, you can read more details in the publication's reference. You can find that you have very prominent clusters of this recruited RNA Pol2. So this here is by super resolution imaging, as referenced by Simon in the beginning. And you can see inside of these boxes that you have very distinct, very, very big, and also pluripotency specific clusters of polymerase that emerge. And these clusters that you can also read in the past work, we and others have found can be explained, or their formation can be explained by surface condensation. So you would have genomic elements like super enhancers that serve as so-called condensation surfaces. The recruited Pol2 and other transcription factors condense on these surfaces and then form these surface-associated condensates. The actual transcribed gene is excluded, as described before, and this can actually really lead to deformations of these condensates. What we can also see, and we as in the lead, two PhD students from our lab, Hamide and Irina, is that you can actually reconstruct from fixed cell images. If you use the so-called pseudo-time reconstruction approach, you can actually reconstruct how a gene visits such a cluster. You will see in the images that I will display one after the other, magenta and always placed in the center, a so-called transcription factory, or a condensate containing high concentrations of these phosphorylated Pol2 that is recruited, and labeled in green by oligopane fluorescence in situ hybridization, the embryonically expressed gene KLF2B. So this is always here on the top right, in this case labeled in green, and here is the cluster that I introduced just before. And by sorting along this pseudo-time coordinate S that you can see labeled above the images, you can see how the gene is step-by-step approaching this cluster until finally it actually gets in touch and gets sort of dissolved inside of the cluster and really comes in contact. And only then it undergoes transcriptional activation and departs again from the cluster. So you can see the cluster itself is heavily perturbed in its organization, so it kind of flies apart or splits even. And the gene is departing step by step into a further distance away from the cluster until finally the cluster itself is relaxing and taking on a round, stable shape, if you wish. So on the left-hand side, you can see this visually explained. We also can actually take hundreds of observations of a gene interacting with the nearest neighbor cluster and see how the gene actually undergoes an approach to the cluster, then departs again. And only once it hits the cluster here, it gets high levels of the recruited RNA polymerase II, and shortly after this actually turns on the transcriptional activity here as it departs the cluster again. You can see it in these plots that sum up the hundreds of observations, or you can see here in sketches how a gene gets induced, associates with its cluster here, gets activated, and only as the RNA is being produced actually detaches from the cluster again and goes on its way away from the cluster. So this gives us already an initial takeaway for the introduction, which is how some embryonic genes are activated by visiting these chromatin-associated clusters. Importantly, this doesn't apply for all of the embryonic genes, but for those we investigate, there are some that behave like this. You initially have the gene here inactive, the promoter region and the gene body, and the super enhancer that holds this cluster or condensate here in the sense of a surface. As the gene is getting induced, the promoter region that you can see here gets a tiny droplet of this cluster also associating with it, then keeps on diffusing and finally hits this cluster. Because two pieces or two droplets, condensates of liquid touch each other, they actually merge and become one sticky glue component that holds these two genomic elements together now. And as the gene undergoes pause release, the PAL2 actually gets activated and can produce RNA transcripts here. The cluster is already getting perturbed as the gene tries to push out of this until finally it here detaches, transcription runs full force, and the gene, as said, goes on its way. This is called, if you do not consider the condensate, the hit and run model of gene regulation at enhancers. If you more specifically consider condensate, it's called the condensate hit and run model. If you want to know more and take some more time to engage with this concept, I highly recommend this review paper here. For this project I'm presenting today, the question is how this model of gene cluster visits actually can apply to genes that undergo transcriptional regulation. So as we turn on genes and we turn off genes, how is this control embedded in the cycle of interaction of a gene with the cluster? And as for many questions, if we kind of need some orientation and just input, we go again to the zebrafish embryo, as it offers, of course, over the course of development, many changes in transcription levels. So we would take here the sphere stage that we have gotten to know before that is more or less identifiable with embryonic pluripotency, a preceding stage, oblong, just 0.3 hours before, 20 minutes, where we still have genome activation as part of development. After pluripotency, we also have first steps of cell lineage specification, so we can expect that certain genes also will be upregulated and certain genes will be downregulated. So I think this should be a very promising setting to see really changes in transcription level. And because we're around pluripotency, we should also expect clusters that the genes might be interacting with. We now deploy the same imaging and labeling approach as we have deployed before, but for 8 genes over these 5 consecutive stages. So what we find here is for a couple of example genes I will first illustrate and explain. We can quantify over the 5 stages how the contact frequency of the gene coming to the cluster and touching it, coming close in the images, changes over development. You can, for example, see that the gene FOXD5 here, over the 5 consecutive stages, loses contact quite dramatically. This is associated with a loss of recruited PAL2 at the location of the gene. So we detect the gene and quantify how much recruited PAL2 is present at the location of that gene. And you can see that nevertheless, the transcription levels are not changing very much. So how to read these graphs? One on the y-axis corresponds to the nucleus background. So this is elevated recruitment. And here one corresponds to the background of the nucleus. So this means there is a bit of transcription going on. You can see that it's not just the method giving you a decrease over time, but other genes actually come into contact, they start to recruit, and this also leads to higher levels of transcription. So it seems to be sensitive. The genes seem to behave differently as well. You have genes that behave very differently from this. So we call them non-contact or non-visitor genes. They have very low levels of contact, relatively low levels of recruitment, but nevertheless are transcribed. You can find even crazier, if you wish, genes like DRLL2 here. We have a bit of cluster contact, but really high levels of recruitment that are transient. And also this seems to somehow have an effect on the transcription levels. It's quantified in this third row here. So you can see there's already quite dynamic behavior, and our hope is fulfilling that the gene transcription goes up and down. We had 8 genes, so there is really a ton of information in here. And our feeling, and probably your feeling as well, is being completely overwhelmed by this. There's so much information that we kind of need to find a system to make sense of this. So we took 3 months to half a year to maybe find a system in this. You get like 10 seconds to jump to the next slide. So here's the system we found. We found that you can actually, if you put it in the right perspective, we can take all these observations and put them on what we call a contact map, and observations actually become consistent with the system. How to read this map or how to draw this is that you take here on this axis, on the x-axis, you place the level of recruited RNA polymerase II at the gene. You place here the level of elongating or transcribing Pol2 on the y-axis, and this allows you to place every gene in every stage at a specific point in this map. So for example, here's the oblong stage of DRLL2, and you can place this here. And as you follow through development, you can see this trajectory developing. You can do this for other genes like FOXP5 as well. You see here how over development we start here at oblong, and we end up in 50% activity at the end of this development time course, if you wish. Additionally, in this map, you can indicate the contact frequency, so here contact percentage by color, meaning that here yellow color corresponds to 30% contact frequency. This is also the highest contact level that you have seen in the slide before. You can see how overdevelopment here turns green and ultimately blue, so you're losing contact over the course of development. Now we have 6 more genes that we want to add to complete the chaos. So if you bear with me for a second, you can still see that there's a system to this. So we have the highest contact frequency here. We have somewhat lower levels of contact here. Then here it turns more blue, but not entirely blue. So we're kind of in this range. And only here in the bottom left do we find really blue values of color. So what this is telling you that there is a systematic map that is emerging. We're using these 2 coordinates, the level of recruited Pol2, the level of elongating Pol2. You can place genes on this map. And depending where you are on this map, you have very high levels of contact, intermediate levels of contact, or almost no contact with Pol2 clusters. So what we take away as a system is that we have genes that can perform as visitor genes if they fall into these 2 classes. They can be either transcribed or not, but in all cases have contact with clusters, except when they become transcribed, the level is somewhat lower. And we have genes in specific stages that behave as non-contact or non-visitor genes. They can be transcribed to some level or not, and they do not need contact for this. And genes can even travel in this map, but still will adhere to this map. Involved in this were people from Vasily Zavrodav's group in Erlangen for the theory and image analysis part, Tim Klingberg, Irina Varta and Aga Pancholi, and yes, Tim Klingberg here, and Vasily, the man himself, over here. In our old lab space, we moved by now, but this is the team who made this possible. Yes, so we have this contact map. The question is, can we get this contact map not only for 8 genes, but actually have an idea if this applies genome-wide or not. And in this case, for once, I will not go to microscopy, but actually to bioinformatics and sequencing-based data. Not me, but actually Aaron, PhD student from our group, who's working a lot on sequence-based bioinformatics, amongst other things. And what we did here was we basically pulled existing RNA sequencing ChIP-seq data sets off the internet out of previous publications. And at first, we're just trying to get whether the microscopy results correspond to what these data are telling us. So what we can do is try to reconstruct the same map by taking the ChIP-seq signal at the genes for the recruited RNA polymerase II. So this gives you Pol2 recruitment. And if we pick the right genes out of the whole embryonic gene set, we can take for many of these genes the RNA signal to more or less directly represent the transcription activity of the gene. And we can then, on this map, take the microscopy assessed genes that have high and low contact and use these data from sequencing-based experiments to see whether the genes end up in the right regions. So the first thing is we will look at the low contact genes in one specific stage. It's the whole set over here. So the low contact genes in microscopy, hollow circles, ended up left of this line, which is the realm of low contact genes. That's what we want. High contact genes that can be assessed in the stage FOXD5 and ZGC64022, they do end up here on the side of visitor genes, as we know from microscopy. So where we can check this, there is perfect correspondence. So we can now go ahead and use the same method to not only assess the genes that we did by microscopy, but basically all the genes that can be assessed in the stage by sequencing-based data. What we find is that inside of these quadrants, so here visitor active and visitor inactive, we find the highest contact frequency for the genes that are visitor gene, or in the quadrant for visitor genes that are inactive, they have the highest contact frequency from high-C data, which we also would expect based on our previous results. As we go to genes that have higher levels in RNA sequencing data, the contact frequency goes down somewhat, so it turns green, but not all the way. This only happens here for genes that have low levels of Pol2 recruitment and different levels of transcription, and these have the lowest contact frequency, just as you would expect. So as far as we can analyze from these data, this actually seems to hold genome-wide, and not just for the genes where we did microscopy. You can ask more things based on sequencing data about the characteristics of these genes or their promoter regions. And what comes out is that those promoters are like small versions of super enhancers, epigenetically speaking. So what you can find is if you assess the chip sequencing data for histone 3 lysine 27 acetylation, that doesn't mean much. This is the mark that tells you this is an enhancer control element. You can find that the visitor genes in yellow and here in green have much elevated levels for this enhancer mark, and the non-visitor genes have actually quite low levels. Also, you can find by ATAC sequencing, which tells you how physically accessible the chromatin fiber is or how unfolded locally. You can find again that the visitor genes are actually quite physically accessible to interacting molecules, while the non-visitor class is not very accessible, which is again very similar to super enhancers that are active. You can find that here the histone 3 lysine 4 trimethylation mark, which tells you whether a gene is not only prepared but really activated in terms of transcription, is here present for the visitor active class and distinguishes it from the inactive, which also makes sense and is what you would expect. And finally, we can find here a pluripotency pioneer factor, a transcription factor that also goes to super enhancers to be quite elevated for the visitor gene class, but not for the non-visitor class. So we can really see that there is a characterization on the epigenetic and transcription control level that tells you that the promoter has a bit of a characteristic as the super enhancer element that is controlling the gene. Okay, so this is for the genome aficionados. If it doesn't tell you that much, it's not that important. Okay, so we have learned many things about how we can generalize the behavior, that all this is somewhat trustworthy, etc., how the genes interact. But the question is, can we generalize this? Also, as Simon alluded, into something that comes more out of physics and gives you the general working principles of the system. So this is where we turn to polymer simulations of this enhancer-promoter interactions in a shoebox in a very simplified setting. Again, we refers to the most part to Roshan Pritzak, who programmed and developed the simulations in discussion with me, who is now found gainful employment at Solgate in Vienna or close to Vienna. Yeah, so the idea behind these simulations is that we take, as I say, a shoebox or a very small spatial compartment, and we tether to the left-hand side of this compartment here a regulatory element or super enhancer, if you wish, in a small part of a chromosome. And on the right-hand side, we tether again a little piece of chromosome containing here the promoter that can control the transcription of this gene body indicated in gray. Additionally, we have red particles representing recruited RNA polymerase II that can cluster on these surfaces, forming surface condensates. This is a drawing. If we use a combination of a Python wrapper for the chemical control transitions or reactions and LAMPs for high-performance polymer simulations, put this together, this is not just a drawing anymore, but it comes close to the video that you've seen in the beginning. So we actually get simulations with realistic physical scales, realistic temporal scales that we can then analyze to understand how we can maybe explain the behavior that we see in experiments. We still need to assign an affinity matrix. So of course, for people caring about who has affinity for whom, who is repelling whom, you can actually adjust the model in that respect and see that some of the included elements have an attraction. Like for example, red particles attract red particles, and red particles are attracted to regulatory elements or super enhancers and to promoters as surfaces. Okay, this is a still image. It's getting a little bit complicated, so let's just look at what it does. This is the shoebox simulation. It's running at about 3 times the speed that you have in the real cell, or how we think we should simulate the real cell. And you can see the super enhancer element on the left-hand side now forming what we call a surface condensate. And this leading to an accumulation of the condensating particles, but also a bunching up of the condensation polymer surface into this transcription factory, if you wish. So this is one element, the super enhancer, that is acting as the condensation surface in blue on the left-hand side. On the right-hand side, you have the gene promoter in blue that is now beginning to attract some of the red particles into its vicinity. And attached to it, you have the currently still inactive gene body in red color. We might have to wait a little bit because the gene is still diffusing, but it is now sticky for the big cluster on the left-hand side. And once it attaches there, which probably happens soon, you can see that the gene body turns gray, which means it is actually turning transcriptionally active now. So here you can see the gene is now being transcribed, and because of this loses its affinity for the red particles and is allowed to basically detach and explore space freely again until it now becomes induced, can again pull the red particles to the blue promoter region and is ready to engage with the transcriptional cluster or surface condensate once it comes into its vicinity again. And as we let the simulation run, we will actually see that there are repeated cycles of this, where the gene runs towards the cluster, engages with the cluster, becomes activated, gets pushed out of the cluster, then gets in, it's active again now. Soon it will be inactive again, and then has the ability to recruit a little piece of that red phase and go into the cluster again. Yeah, as you can see now, it has just deactivated and activated once more. So with this, we can reproduce these gene cluster interaction cycles, as you can see. And the last thing that we want to ask is, how can we take this model to learn about how we construct or design, hypothetically design, a gene that falls into one of these 4 different categories? What we have found from assessing different versions and designs of the simulation is that we have 2 really crucial design parameters that are sufficient to explain the whole distribution. The first one is the promoter length. So you can imagine if the promoter is acting as like a tiny condensation surface, if you have a short promoter, you can attract a little bit of that condensate material. And if you have a long promoter, you can have higher amounts of the condensate material, which might take you over a threshold for activation that also allows you to effectively interact and attach to transcriptional clusters that will lead to even higher chance of activation. This is one parameter. The other one is the activation rate. So once you cross this threshold, your gene can, in principle, be activated, but this happens with a certain probability per time or a rate of activation. This is the second parameter that we need. And as we vary these parameters in really an extensive set of simulations, you can see that all these points here populate, again, the same coordinates of this contact map. So we have here recruited RNA polymerase II, as we can quantify from microscopy measurement of these simulations. And on the y-axis, we have transcription activity, which gives the second coordinate. Again, we can put the contact percentage. It's very comparable percentages that you see over here. And maybe from this, you get an idea that over here, the values are higher, and over here, they are lower or more blue. If it is not immediately obvious, we did an interpolation and averaging for you. So you can see here that in the bottom left, we have low values of contact, intermediate levels of contact, and high levels of contact, just as we have seen in both ways of assessing this contact map experimentally. You can also now ask inside of these four quadrants, so here are the visitor genes, the non-visitor genes, transcriptionally active or inactive. You can see that the promoter length for all the gene designs that end up here is short. So you have one monomeric or one monomer contributing to most of the promoter designs, while for the visitor genes, you have two or in many cases, three monomers that are needed to construct the promoter. So you need a long promoter. And the activation rate needs to be quite high for the non-visitor genes, but nevertheless leads to relatively low levels of transcription here. While here, for the visitor genes that come into contact for a long time with the clusters, you find that even a low activation rate is sufficient to activate them to a really high level of transcription. And yes, you can find the visitor genes have high levels of contact, the non-visitor genes have low levels of contact, which confirms just what we expect here. All right, so this is to finish up. Back to the initial question, how do some embryonic genes visit chromatin-associated condensates to undergo transcriptional control? We can bring back this initial picture and add a few crucial details. The first one being that here for the induction, that then allows the gene to interact with the promoter, with the super enhancer associated surface condensate. What we need is a promoter of sufficient length that also takes on epigenetic characteristics of an enhancer. This allows it to recruit this mini condensate that then can fuse with a bigger condensate to make these two things stick to one another. Once you are inside of a cluster, you still need to activate to also be kicked out of it. So the second control parameter that seems to be central is the rate at which the Pol2 that associates with a gene is allowed to go into actual elongation activity, allowing also the gene to finally escape and detach from the cluster again. So that's the two additional takeaways compared to our previous work that you can read here. These are the people contributing to the new work that I presented today. And I would like to give a nod to other lab members who contribute to the numerous other projects that we carry out. And none of this would be possible, of course, if we wouldn't have a few euros to support the work. I've taken enough of your time, so I'll hand back over to whoever is the chair. Thank you for listening.